Hello everyone. Today I plan to give you an overview of the research I have conducted so far towards my portfolio. I appreciate any constructive feedback to assist me. I have expanded my epistemological knowledge of biology to generalize it as the life science. Biology takes on an anti-vitalism approach, meaning that it breaks life into its separate, non-living components to understand its basics. For example, a plant is a living organism, yet photosynthesis, a broken down component of that life, is in itself not seen as living. The communications of findings in biology favour quantitative data that can be reproducible to confirm its validity. The scientific method is the largely accepted method in which scientific data is produced. Using the diagram as a prompt, the scientific method poses a question, researches the already available information, then poses a hypothesis, which is a proposed explanation to your question, which includes the variables that you would like to test. Then you test the hypothesis, analyze the data, and then from your analysed data, you make a conclusion. Either your hypothesis was wrong, so your explanation was wrong, so you make a new hypothesis, a new explanation, and retest. Otherwise, if you were correct, you retest anyway to double check yourself. You communicate your findings and then propose it to the scientific community. The scientific community will have a look at it and either agree with your hypothesis or disagree based on your methodology and your data. From an epistemological point of view, the scientific method in itself is a tautological structure, meaning that the knowledge is built by repeating itself with different terminology and contradicting the previous knowledge if necessary. For information to be considered substantiated in science, it needs quantitative data to deem the hypothesis true or false. After my original meeting with Brian, I decided to base my pedagogy around course relevance. After researching relevance as a pedagogy, I found there are different types of relevance, even the teacher's mannerisms and persona in the classroom and how the students can relate to that. After researching the method, I honed in specifically on outside course relevance. This method is choosing parallels between the content and events, interests, or desires that lay outside the classroom. Using this pedagogy has resulted in an increase of student engagement with the content and a greater motivation for students to learn. With the inquiry plans in mind, I decided I also needed to find a method to structure my lessons. To do this, I have turned to the 5E framework. The relevance topic will be used in the engaged stage of the framework, and the 5E model is based on a constructivist design to increase the metacognitive process of the students while they're in the classroom. I have chose to use the topic of genetics for my portfolio as I felt this was my weakest link. I have decided to specifically use the mutations topic within the genetics unit. First, I looked at the terminology to see if there was anything I didn't understand and turning to my textbook for clarification. Mutations occurring during the DNA replication are classified into two types. The first is a point mutation, which alters a single base, generally by swapping the base for another. There are two types of point mutations, and this is a myosin mutation, which swaps out a single guanine for a single cytosine, creating a single mutation within the protein strand. The second is a nonsense mutation, where a single base is swapped out for a stop codon, which truncates the protein sooner than what it was supposed to be. The next type we have is a frame shift mutation, which is more severe and is when an mRNA code start point is shifted along and translates to, so the code translates to an amino acid, and when it is shifted across, the code is translating to an incorrect amino acid to what the mRNA originally intended, which is more harmful as it creates an entire sequence of incorrect amino acids. Mutations during cell division is simply somewhere in the process of meiosis, the chromatids have not been divided equally and is called non-disjunction. So here is a normal sequence of DNA and everything is divided up equally, and for non-disjunction here is two separate examples, but essentially it is when the chromatids have not been divided up equally into the cells. So for example, this cell here has ended up with 3 chromatids and this ended up 2, which then translates into the gametes. So these gametes have 3, these only have 1, and here's another example of how it's been divided up inequally. The gametes are your sex cells, so therefore it'll have devastating consequences for the offspring. All mutations can occur from exposing DNA with a mutagen, which can range from sun exposure to chemicals. Now that we have these 3 types of mutation, and they make up the base information for the entire topic. Now that we have the basics, we need to dive a bit deeper and understand that non-disjunction leads to aneuploidy. Aneuploidy, I had no idea about, so doing some research, ploidy is the number of chromosomes in the cell, which for a human is 23 pairs of chromosomes. And if this figure is abnormal, then it refers to aneuploidy. And non-disjunction, which we've already gone over, the unequal distribution of chromosomes during cell division, can lead to aneuploidy. So this particular karyotype is Down syndrome. And in Down syndrome, on the 21st pair of chromosomes, there is 3 rather than a pair of 2. Now, as I did a little bit more research to expand my own knowledge outside the scope of the curriculum, I was looking at the procedure needed to test if the embryo has aneuploidy. I found out that as women age, so does their genetic integrity of the eggs, and at the age of 35, women in Australia are strongly encouraged to get an invasive procedure called amniocentesis. It was interesting to find out that there are less invasive tests available but are deemed less cost effective, so they are less accessible. Okay, so here students need to use a human karyotype to identify ploidy changes and predict a genetic disorder from given data. So here is one example of a karyotype, and they need to identify the ploidy changes. So from here, as we've gone over, we know that there are supposed to be 23 pairs of chromosomes, but as the student should be able to pick out, on the 23rd pair there is only 1. So we can do some different activities with that. Okay, next point. Describe how inherited mutations can alter the variations in genotype of offspring. I could not think in specifics any inherited mutations off the top of my head, so I researched and found that blue eyes originated as a mutation to the OCA2 gene, preventing pigmentation of the eye. It is a recessive mutation, meaning that offsprings need to have both recessive alleles to display this trait. As we can see in this first diagram, looking here, mum has both, which increases the offspring's chance of receiving that genotype. And then doing a quick cross, I'm hoping this is bringing back some memories of Punnett squares, like that. 50% of the offspring received both recessive alleles to display that trait. So that is how That is how offspring will show variations in the genotype of this particular mutation. This can also occur in other mutations such as cystic fibrosis. Okay, as a note here, students need to be able to understand a large number of chemical mutagens that are carcinogenic and interact with DNA. An example I found during my research was Agent Orange. This is expanding outside the scope of the curriculum itself, but the use of Agent Orange in Vietnam War was caused many birth defects in children whose parents were exposed to the chemical. And the agent contained dioxin, which is a highly toxic carcinogenic. So that's just one example I found, but there are many out there. I ran out of room on the slide, so here's the content references that I've used for this presentation. To merge my content with my chosen pedagogy, I have incorporated relevance by using examples of mutations that they may have seen in people of their community. I'm hoping that with a greater education in connecting textbook mutations with real people, that students will see the relevance of learning this content and create a greater air of acceptance. I have divided the content into 3 lessons, given the 3 notional hours. In all the lessons, students will need to learn the basic syllabus as well as answering the overarching relevance question, why are mutations relevant in our community? In the first lesson, I was thinking of engaging them, the first part of the 5E framework, to look at their arms and see if they can find a freckle. Then opening up discussion on sun exposure and how it mutates our skin cells on a DNA level. This will lead into the explore stage of the 5E framework and continue throughout the lesson. In the second lesson, as it relates to non-disjunction and aneuploidy, I was thinking of guiding students to this site or a printout of the site, as there is various disorders on there. And first they will need to answer specific questions of the karyotype, the disorder and the genetic features before turning to how this affects the individual. This will be a part of the explore stage of the framework. They will then need to explain what they have learnt and so on, following the structure. The third lesson, I'm a little bit conflicted and would particularly like some feedback. I would like students to complete an activity regarding an autosomal recessive mutation where they need to predict the variations in the offspring. I've considered the blue eye example as it is widely found in the community, but after more research, I found this mutation is more considered a trait in humans as it has been around for so long rather than a specific mutation, though I'm happy to take on feedback. As a plan B, I've found these other hereditary mutations and wanted to hear what people thought about which ones students might find more relevant: cystic fibrosis, sickle cell anemia, or juvenile glaucoma. Any feedback is welcome. To extend my knowledge as a professional, I have turned to the APSTs. I plan to implement the constructive feedback from my colleagues that I'll receive from this presentation. This will build my skills as a teacher and my resilience to take on feedback. To engage in professional learning, I plan to extend my knowledge through ongoing research into the fields of my teaching areas to stay up to date with the fluidity of the scientific knowledge. Also note that due to the time restraints of this video, I have only mentioned a partial amount of the research that I have conducted to go towards the portfolio. To apply the professional learning, I plan to participate in workshops that are readily available to me to build my skill set and use these skills in the classroom. Thank you everyone for listening to my presentation, and I appreciate any constructive feedback that you have for me. Thanks again. Bye.